is that right that you could see some IL-17 signature in the T cell? So, and luckily this is TFH. So if you only do CD4, you may not see it too well. But now with TFH, we might see better. And you can see that these are B cell, and then this one is plasma in each sample. And then this is T cell in each individual. Then this is the Th1 signature. And you could see a little bit higher in the lupus compared to the, in a way I was surprised. Not like this is so much higher. It's a little bit higher in lupus compared to healthy. And you could see that the Th17 signature is also higher in the lupus compared to the healthy. And this gives you a larger view so you can see better that they have more Th17 cell in lupus versus healthy. So with that, and of course,
you know one thing, Th17 is CCR6.
CCR6 and then another marker.
Right. I think it's
yeah.
I was just wondering, Mingao could, now that you show it like this.
Right, she could do it. Yeah, of course we can zoom into. Right.
Yeah, gate on C,
right.
or actually CT could be good at this. Gate on both CCR6 positive and the other, because two chemokine receptor genes define, you know,
Th1,
I know.
Th17. Yeah, that's right. All right, so with that, we can propose a very similar model now for lupus. What's that mean? Of course, it's not virus stimulation, but it's elevation in type 1 interferon. And then also we know in germinal center with BXD2, there's an IL-17 signature there. So what happened is here, it's the same. It's just the same thing, except it's not viral NP, it's an RMP antigen. So again, that RMP antigen keeps stimulated through this TR7 endosome with the type 1 interferon amplification that these B cells, yeah, they generate tons of these B cells. And these B cells extrafollicular, DN2. But at the same time, these B cells make a lot of interferon. Well, I shouldn't say a lot. Compared to PDC, it's less, but they make their own interferon to stimulate neighboring B cell survival. So a lot of the non-RMP B cell in lupus, I'm sure there are a lot of MMDA, histone, those kind of antigen, not the RMP component. So these B cells get the advantage to be stimulated by interferon, and they survive. Maybe they go into the germinal center that they are stimulated by IL-17, and this is a follicular. Of course, it could be IL-21, even IL-4 even, and they are the follicular and germinal center pathway. So you get a lot of both kind of B cell generated in lupus, but this is always the dominant one. This is the dominant one, you see a lot of them, but so they make this antibody, whether they are pathogenic, we are trying to figure that out. But the other kind, like NMDA, the other kind, they could be pathogenic too, and you could find it in lupus patients. So this is the model for lupus now. So we could submit a new R01. We already have an R01. We say we could make minimal modification and just submit it. So we can do that. And meanwhile, work on this model and then maybe even submit another R01 in the future. So we potentially can submit it to R01 about lupus. Just based on
one thing that I'm thinking of is the role of IL-4 receptor.
Right, IL-4 is downregulated. So even in COVID-19, it's downregulated. Right. So that downregulation is, I think that's somehow that is a signature for naive resting B cell. And I think type 1 interferon, IL-4, they both counteracting with each other. So I think that downregulation is enabling this type 1 interferon signal pathway, or TR7. So that's needed for TR7 signal to go through.
But the question was, as you recall, Not so much which is chicken and egg. I mean, yeah, it's a
Right.
simple.
It's hard to say which one first. That is a very.
No, that's not the important question. Although the viewers,
right. That's right.
To me, that's not important.
Right.
It's the important question is why is IL-4 receptor high or not? And we got around it. We know. I asked Finkelman, IL-4. He discovered IL-4. He says the main regulator of IL-4 receptor is IL-4. Okay, so we know why it's high. It's IL-4 is high or not. No. So then, okay, so why is IL-4 high or not? So then you get back to the following, which is a sort of an epidemiological question. IL-4 is high to protect you from, helminths and worms, and they make mast cells, and it's an allergy sort of a response. So here's the problem: is if you either get virus, you either can get yourself protected from viruses, or you can protect yourself from the worms. And you looked at the map of the world. Remember how perfectly the distribution of SLE matched the absence of helminth infection.
Right.
So strongyloides, whatever it was. Right. So it's quite interesting that the reason why IL-4 receptor is high in, well, okay, then we go back to the other thing. It's black versus white. White folk, the Africa, we clean ourselves up. We went up north and no worms. So then we're sitting around and no worms. So we don't need wait a minute. If we don't have any worms.
Well, I still think that IL-4, it definitely has a way to block the TR7. So I think I always thought those are the TR7 tolerized B cell. So, but for the protection of cells, so immune system, you always say so, that the first thing you gotta do is not to react to cell. That's even the first principle of immune system. Not even to antivirus, antipathogen or not. The first principle is not to react to cell. So I think that the IL-4 receptor, those IL-4 receptor positive cells being tolerized, so they won't react to this RNP antigen. So that's the prevent the immune tolerance loss to self-antigen. That is the marker for that.
Well, it's a break.
Right.
It's a break. You put the brake on the whole thing right from the start. But then why do some people have the brake and some people don't?
I think that then interferon somehow, if this signal too strong, the TR7, they lost the TR7 tolerance. That because they have very strong interferon beta, or they have too strong RNP antigen stimulation, that can break that tolerance. So that's the thing.
But I think we showed in the first time of the grant that patients who had lupus had low IL-4. We even found it once. Remember, SLE patients had low IL-4.
Right.
That's been reported in actually by 3, maybe 4 papers in the whole world now. Because nobody those patients have high everything. They have high interferon, they have high IL-1, they have high TNF, they have high they have high IL-17, interferon gamma, they have high. But they don't have IL-4. IL-4 is never high. What's this? So they leave it out of the paper. Well, let's not report that. But the point is IL-4 is low in lupus, and that's an independent pathway.
We don't know which way is first. That's always a question. Yeah.
No, but IL-4 is the #1 regulator of IL-4 receptor. So IL-4 is low.
Yeah, but so we don't know which way. Yeah, is it because interferon high, so IL-4 is low, or what? That's always tricky.
But IL-4 receptor is low. Why would interferon turn off
IL-4? Just the whole parallel, both innate, everything. So they all always go parallel.
I mean, we know it's not that complicated. It goes back 20 years ago when T cells became Th1 and Th2. Now the Th17 and so forth. But Th2s make IL-4.
Right.
So it's really, when you get to thinking about it, it's the T cell that did it. The T cell makes IL-4 or not. Now you're saying, so that's out there. And now you're saying, depending on what it did, then the Th1s make interferon, is interferon gamma in here? Anyway, it can help, but it starts with the T cell, and that starts with the worms. All right, but okay, so I think it's still a question of, you go back, no matter how far back you go, you go back one more step.
Right.
All right.
All right, so with both COVID-19 and lupus, we're gonna study almost identical thing, except the antigens are not the same. So that's the only difference. Otherwise, what we are doing, it will be completely identical. So being able to find how to tag the antigen reactive B cell is the most important, is the missing link to enable us to answer this question. But with the lupus, yes. So the plans I have, I need to finish the IL-4 receptor manuscript with Mingao. And then we should do a more careful analysis of TFA transcriptome in lupus. Then we should really work hard to get this liberal sequencing strategy to work, so that this will allow us to study both lupus and COVID-19. So I think the steps to do is determine. So of course we're gonna get the peptide. It's already what is a fluorochrome, and then it's a peptide with oligos, everything together. So, but we first need to know the peptide that we think it can bind to the B cell receptor. Would that really enable us to identify the B cell or not? So
the Libra seq was SM, right?
Well, so we have these three. We ordered these three.
Yeah, right.
Right. So with Sharon's help, we designed these three: the Smith D3, Histone 3.1, and then U1SMP. So these are the three antigens we have. So of course, first thing is to do flow cytometry, see if they bind or not. If they do, we isolate those B cells and stimulate them. Would they make the correct kind of antibody response or not? Then if all these can be confirmed, then we use this. Then the third thing, can they really work with a single cell? I think with Sharon's design, it will work. So I think that, so the first two questions are harder. That is really, they would they bind to the B cell, the right B cell or not? Those are the more challenging part. Once those are verified, then the single cell, I think it's gonna work. So those are the things. This will be the key experiment for us to do. So that's the experiment we should push hard for.
One thing I was thinking of, if you go back up to that thing, let's take SM. It binds anti-SM.
Anti-SM. And right. The SM is that RG component. Right now, with the COVID-19 even, I realize how important this part is, because this is exactly the part that binds to the RNA. Because we always wonder why this is such a highly positive charged region. It's sort of defined on what we previously think, because previously people think about lupus autoreactive B cell, they're thinking about B cell that binds to DNA. So they think the CDR3 should be positively charged, right, because they want to bind to DNA. Whereas this Smith is not. This is so the antibody, the antigen itself is positively charged. So the CDR3 should be negatively charged. And you say, and you always say, this is such a funny sequence. Why is RG? And this SDMA means symmetric dimethylated arginine. So that's what it means. So it's still arginine, but it just somehow it's methylated. But it's still, so it's RG, and you say, well, that's such an awkward, what kind of sequence? When that binds very unspecifically to everything, as it turns out, this part is absolutely needed to have the best binding to the RNA. So you can imagine why why the RNA virus like to use, maybe they have very similar component, just like the SM. And that's the, of course, not the same sequence, but the idea is that MP is just like the SM that binds to the RNA during the translation for that purpose. So, yes.
That's good. I mean, that's a good thing too, to say that.
Right.
The reason why I was just thinking of this was I believe that trying to think of how this was. Tongzhou purified SM.
Right.
Am I wrong here? Remember, we get a bunch of SM that use purified SM. What did he purify the SM on? He purified them on a column of anti-SM.
The column was, I think, was the anti-SM, right?
Or I think it was SM itself.
Right. So he first purified the SM antibody. Then he immunized the mice with the anti-SM. Then he just, so it's So it's an antibody against antibody. Right.
Right. The point I'm getting to is, then he purified it in a column. Whatever he used will recognize SM, which is this thing. I mean, I wonder if we can just.
I don't know if it will be the same, because that SM could be SMD1, not necessarily D3.
Yeah. Well, at any rate, we didn't give him bad stuff. I mean, it could be a few things, but the point is that it might be hard to show that this binds to cells. But if we had those beads or whatever he uses as a target for this purified SM, it should bind to that versus.
You mean a double staining?
Yeah, just do something. We know what these antigens are. So if these things, if we got antibodies that bind to this, like anti-SM will bind to this, right?
Yeah.
So but whatever he used, anti-SM would bind to that, anti-SM binds to this. Somehow put those together as a nice experiment so we can demonstrate that anti-SM binds to this.
Yeah.
I mean, why? I mean, it's like we could even do, we got purified anti-SM. We could almost do like just a simple ELISA assay, coat a plate with some of this and show that SM binds to it.
Yeah, that's true. Yeah.
Right? Do something simple to demonstrate it works.
Well, that antibody, that's an anti-SM. So it's.
Well, then.
Okay. So that means you coat the plate with this thing and you incubate with the patient's serum. Then you add Tongzhou's antibody to see if that.
Oh yeah, something like that.
That's something like that. Right.
Right. Yeah, that's even better.
Right.
Some way to do it so that you show this works.
Right.
And before you get into cells, because then you get into other problems.
Right. Something like that. Right. So this is the thing, but I guess finally we solved the science part. Now it's the how we're gonna do it. That's why it finally leads to this thing. What do we need so we can safely work on human cells? Because for both lupus, COVID-19, we need that. But I think we already discussed. So Monday, 3 o'clock, let's meet in the lab so we can identify how we can set up everything so we can do it. Yeah. So I think that's the end of this discussion. Yeah. So we kind of want to do it, but I just don't know how we can do it. This is at that stage. Yeah. All right. So it's good that we talked about it. So yeah. All right.
All right. That's very good.